Hey y'all. So today we are going to do kind of like a Unit 4 and Unit 5 little review. Um, 4 is really not my favorite with all the cell communication stuff. So like I kind of focus more on the 5 side of it. Because Unit 4 is just it comes up in all the sections. Go ahead and type in the code into the game so you can kind of get entered. I can't play the game until there's actually someone in the game. And then after you get in the game, if you'll type the code into the question box, so I'll be able to pin it. So again, go ahead and just type your game code in so you'll be able to join the game. If you don't want to join the game, that's perfectly fine. You can kind of just answer within the chat. But if you don't get into the game, I can't even start the game. So I need at least one person to get into the game. And this is kind of just how we run it. We'll play the game, and then as we play those games, we'll be able to go over all the answers, and we can kind of answer any other questions that you have about this information, so that you are on the right track to success. And as of right now, no one's in the game, so I'm just gonna kind of wait, because I can't go into the game unless you're in the game. But of course, I told y'all 4 o'clock, and it's a little bit early, so just gonna kind of wait. How are y'all doing today? Anybody got anything exciting going on? Tomorrow our school is celebrating Pie Day, well, at least the faculty is. So we were all supposed to make pies, and so I don't really like pie, so I made a trifle. So that's what I just did a second ago, is I went and laid out all these little cups, and I made like mini trifles for the whole faculty, so that we can all have some yummy treats tomorrow, which probably we're going to be eating the treats like all week long, because there's so much pie being brought to school tomorrow that it's going to be interesting. So no one has logged into the game, so I can't play the game without you. So if someone wants to log into the game, that would be helpful. Again, I'm going to kind of probably kill a little bit of time till like 4 or 5, just to let anybody who's planning on joining us kind of join us. And hey, it's Miss Pratt. Hi, Miss Pratt. So again, just kind of type yourself into the game, put that code in. It's 982464. And then after you get yourself in the game, if you could type that question, then we'll be able to get that code for everyone else. You caught a live finally. Yeah, you did. This one's a live that's just review games. So I said I'd wait until like 4 or 5 to kind of start the first game, just to give students a chance to kind of get in. And again, I do see there's one person in the game, so I can play the game without anyone else getting in it. But, you know, it's nice and fun. The reason why I like using Quizizz is that it's going to put the questions onto your own devices. Like, you can actually see the questions and then answer on your device, which is a lot better than what happens with Kahoot. Kahoot's just, you know, the different colored and shapes, and I'm not really a fan of those. If someone will please type the game code into the question box, that would help out a lot for anybody who wants to join the game late. So I'll read that code out, so if someone wants to type it in for me, that'd be amazing. Again, we're going to type it into the question box, that way I can pin it. So it's 982464. So if you can type that into the question box, 982464, I'll be able to pin it. And then I'll be able to kind of keep that pin, so that if anybody comes in later, they'll be able to still join our game. So again, if you could type that in the question box, which my question box doesn't have anything yet. No. But hey, they've given us poll features now. That's pretty cool. Well, if no one types in the code in the question box, then I just won't be able to do that, which will be fine. How's everyone else doing today? Miss Pratt, how's everything going over there? Did y'all get any snow with all the new stuff? Yes. Thank you, Oliver. There we go. Got it pinned. Okay. So now anybody who joins in later will be able to join that game. And I see that Miss Pratt also did it. Thank you, Miss Pratt. Okay. I don't like to tell time correctly, so it's 4:02, and we got some people in. So I figure we'll just go ahead and start the game. So we're going to press that start button, so we'll be able to play our game. And hopefully I've got it all set up so we'll be able to see. Okay, so phase involving microtubules to separate sister chromatids to opposite poles of the cell. Which phase is that? Okay. So looks like we got everybody with their answers in. So the phase involving microtubules separating sister chromatids to opposite sides of the pole would be, of course, anaphase, because we're going to move them apart. So if you remember our little cell hand dance, you've got prophase, metaphase, anaphase, telophase, and then after mitosis, you have cytokinesis. So that should be anaphase. Great job, y'all. Next question. So phase involving sister chromatids aligning on a plate in the middle of cell. Looks like we've got everybody with their answers in. So that would be, of course, metaphase. So we've, as I said before, we have prophase, metaphase, anaphase, telophase, and cytokinesis. So prophase is when they're preparing. Metaphase is when the sister chromatids line up in the middle. Anaphase is when they're coming apart. And then, of course, our telophase when we have 2 new nuclei. So, phase involving pairing of sister chromatids and condensing of chromatin into chromosomes. Okay. So if we're talking about them condensing, that's of course our prophase when we're preparing for that cell division. Great job, y'all. Yay! Love when I see that green line so tall. I think you guys are rocking this, or you all are rocking this. So phase involving nuclear formation of the envelope around those separated chromatids. Which, by process of elimination, you should be able to easily get this one, because it's ones that we didn't already do. That's of course telophase, where I have a 2 new... Ooh, what happened? Oh, our 2 new nuclei. So of course, prophase is when we're preparing. So that was when we condense those chromatids down. The sister chromatids would find one another. Metaphase, when those sister chromatids line up on the metaphase plate. Now, I specifically say sister chromatids because when we talk about meiosis, it's going to actually be homologous chromosomes or sister chromatids, depending on which phase of meiosis we're in. Anaphase is, again, those sister chromatids coming apart from each other since we're in mitosis. But if we were in meiosis, then that would be either homologous chromosomes or sister chromatids. And then telophase is when we have those 2 new nuclei. Great job, y'all. Which checkpoint involves ensuring all microtubules are attached to kinetochores? Might involve a little bit more thought process here. Okay, so we kind of think this through for a second. So microtubules are going to be extending from our centrioles on the 2 poles of the cell. And those microtubules are going to be attached to these little proteins that are along the centromeres of the chromosomes. And so they're going to attach to pull them apart. And so if I want to make sure that all those microtubules are attached, that's going to be my M checkpoint, because it's right in the middle of mitosis. So that should be your M checkpoint. Look, we got 2 people. Way to go, y'all. Nice job. I don't see anybody in the chat asking questions, so I think we're good. So which checkpoint ensures all DNA has been replicated and there's no errors that have existed? And that no is in, of course, quotes, because errors will exist. But this is talking about like major errors that would be detected by the cell. So unit 6 is actually in 2 weeks. So we don't do unit 6 this week. Oh, why'd the question go? Question disappeared. That's okay. So since we're trying to make sure that all that DNA has been replicated, this has to be something that takes place after replication. And replication took place in the S phase of interphase. And so that would be your G2 checkpoint. Okay, there is no such thing as an S checkpoint. I've literally just made that up as like a distractor for you. So which checkpoint involves determining whether the cell should divide? Okay, so if we're trying to figure out if the cell should divide, it's going to need to get that growth factor, or it needs to get some type of signal molecule, some type of ligand binding to the receptor that then causes the cell to go through that division. So that's our G1 checkpoint, allowing us to start going through G1 and then going into the S phase, then to the G2, and then finally into mitosis. So kind of in the middle of that G1 phase is where we're going to see the G1 checkpoint. I'm on fire. The S phase option, tricky. Yeah, I know. You gotta have those distractors in there, Oriel, right? Okay, which of the following occurs before the process of mitosis? We got 2 people in. Which sometimes kind of people stop playing the game, and that's kind of what happens in these later rounds, is that people have stopped playing the game, and that's why they haven't all voted yet. So which of all occurs before process of mitosis? Well, sister chromosomes pairing up occurs in prophase. DNA being replicated is during the S phase of interphase, which happens before mitosis. Chromosome being condensed down into chromosomes, that occurs also in our prophase. And then cytoplasm divided between 2 cells is going to occur in cytokinesis that happens after mitosis. So it should be DNA is replicated. So which of the following describes the order of processes in the cell cycle? So again, this is cell cycle, not just mitosis. Okay, so this is some acronym that some people come up with is IPMATC. So it's going to be interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So IPMATC. So there's been a couple different ways people remember that, but that's kind of the big one that people use. So next question, which I believe is our last question in this game, which we have 3 other games that we can play. Y'all will tell me which one you want me to do next. Which one describes the result of mitosis? So after we go through mitosis, what is the result? Mitosis is nuclear division. So someone in the chat just said, what is mitosis? Mitosis is the process of nuclear division where you're dividing up the nucleus of a cell. So it says, which of the following describes the result of mitosis? So after mitosis, we're going to end up having 2 cells. So that allows us to know it's either this one or this one. And those cells are going to be identical. And they will be diploid because they're going to be exactly like the parent cell. The goal of mitosis is to make identical copies of that cell. So they will be 2 identical diploid cells. And that, I believe, leads us to the end of this game. So that was your last question. So do you want to exit? Of course you want to exit. Okay, and I'm sorry about it being so white. I can't control that. Okay, so the other games that we have as options, we can do cell communication, we can do meiosis, or we can do heredity. So whichever one you want to do, it doesn't matter to me. Please just kind of vote in the chat which one you want me to do next. So we could do cell communication, we could do meiosis, or we can do heredity. Those are the 3 options that we have for our next game. So do we want cell communication, meiosis, or heredity? Please type it in the chat which one you would like to do next. So cell communication, meiosis, or heredity. Anyone want to join in? Tell me which game we're doing next. Anyone? Y'all are the leaders of the game, so y'all tell me what you want done. Hello? Am I by myself? Cell communication, meiosis, or heredity? I think I'm by myself again. No one wants to answer me. Okay, so I guess since we just did mitosis, then I guess we'll just go over to meiosis and play this one because no one's answering. So let's give you a moment to be able to type in the code and get that white crazy screen off. Okay, so let's see if I can blow that up again. Okay, so there's your code. 922 499. So the next code we have is will be for meiosis. So we do 922 499. So go ahead and type that in so you can get into the game. And then if someone will type that into the chat, so we'll be able to keep the game up, the code up for the game. Hey, John from Market Learning. Well, I'm assuming it's John. We're just playing review games over here. So if someone will join the game, 922 499. And then if they will also put it into the question box so I can pin it for anyone who comes later, that would be amazing. Again, that code is 922 499. And we're doing a game on meiosis. If I can't press the start button unless there's someone there, so it won't actually let me do anything. Hey, look! Miss Pratt did it for me. Thanks, Miss Pratt. You're the best. 922. There we go. Okay, so we got that in. We still have nobody in the game, but hey, at least we got the code so that if anyone else wants to join, hey, we got Mulberry Moo. I kind of like this thing that like auto generates your names for you. Then there's, confidence. No one really knows confidentiality. No one knows who you are. It also decreases your ability to type mean things on my screen. Ha ha ha. Okay, so there's only 2 people in this room and only 1 person in the game, so I think we're just gonna go ahead and start. Which I think I have to change the size of the font again on my screen, so you'll be able to see everything again. Should be wrong size again. There we go. Okay. So what is the result of meiosis? No, that's not the right size. That's what we need. Okay. So what's the result of meiosis? So in meiosis, we just looked at mitosis, right? So mitosis, we were getting 2 identical diploid cells. So in meiosis, I'm trying to make the gametes. The gametes are then going to go through fertilization, fusing together to create a new diploid. So in order for me to do that, they have to be haploid cells. So that tells me it's either this one or this one. And then we also go through 2 rounds of division with meiosis. So we're going to have 4 haploid unique cells. So that means that they're going to be genetically unique due to crossing over and independent assortment. Nice job to the 2 people playing. So when does crossing over take place? And as a reminder, crossing over is when those non-sister chromatids exchange their genetic information. So when does that take place? Okay, so crossing over actually takes place during prophase 1. So when you're going through that condensing process and our sister chromatids are pairing up, we're actually seeing now the whole homologous chromosomes will pair up. So they make this little tetrad. And so in the interior chromatids, one from maternal, one paternal, they cross over and they exchange that genetic information. That's the process of crossing over. So that's what happens in prophase 1. I see there's a question in the chat. I'm going to get the next question up and then I'll answer your question because I haven't actually read it yet. But I was giving a second for this to pop up, so we'll be able to see that while I answer. It says, I'm taking AP Bio next year over the summer. Do you have any recommendations for AP Bio books? Okay, so in terms of to get ready for next year, I honestly think you need to take your summer and just relax. You should have plenty of time next year to go through all your biology stuff. So just enjoy your summer and don't worry about AP Bio. If you absolutely 100% have to do something, then you can maybe go grab like a Barron's book or a Princeton book and just kind of do some casual reading through it. It'll allow you to have a foundation as your teacher then lectures and teaches the material next year. But again, I do not think you need to do all that. You could just enjoy your summer and relax and be a teenager. So the question we had on the game, when does independent assortment occur? So independent assortment has to do with when those homologous chromosomes independently align on the metaphase plate. And so since we're still looking at those homologous chromosomes, that would be metaphase 1. So metaphase 1 is what the answer is for this one. Ooh, we only got 1 person and they didn't get it right. Miss Pratt, what are you doing? Okay, so during meiosis 1, which of the following are paired, aligned, and separated? And if Shy is, if Shy is still in here, I'm planning on definitely running this as much as I can next year. I'll probably have more stuff posting, you know, as the year progresses. You know, it all depends on whether I'm teaching it next year. I don't know yet if I'm teaching AP Bio. They haven't decided. You know how administration is. They change our courses last minute sometimes. So during meiosis 1, which of the following are paired, aligned, and separated? So in terms of pairing up, that's going to, of course, be... we paired them up during prophase, but then when we talk about them being aligned, that actually had to do with metaphase, and then the separation actually has to do with anaphase, right? So what thing is allowing for all this stuff to take place? That has to do with those kinetical microtubules. Those are the things that are going to kind of like do that moving around that we see during meiosis. Oh, wait, what did I talk about? Oh, which of the following? I was completely misreading that question. It didn't ask us what did it. So they're asking what thing is being paired aligned. So that's your homologous chromosomes. Remember, there's one from your maternal, one from your paternal. And so they're just saying what's getting that pair, that alignment, and that separation. I'm sorry, I misread the question because I was too busy talking about other things on the chat. So during meiosis 2, which of the following is paired, aligned, and separated? So now we're asking during meiosis 2, what is being paired up and what is being aligned and what is being separated? So now I'm reading the correct question. This is what happens when you rush and try to eyeball a question instead of reading it. I can hear my son just woke up from his nap. Okay, so instead of our homologous chromosomes, now we're looking at our sister chromatids, are the ones that are actually being paired and lined up and all that fun stuff. Nice job, y'all. So how does mitosis and meiosis differ? So this was actually a free response question a couple years ago where they asked you to talk about comparing and contrasting mitosis and meiosis. And when students saw this question, they forgot to mention both. So make sure that whenever you see a compare and contrast on the AP exam that you actually mention both things. Like in mitosis and meiosis, this happens. In mitosis, this happens. Versus in meiosis, this happens. Kind of make sure you're bringing both those things up. So mitosis is going to go through one round of replication. Versus meiosis is going to... Oh, actually, they both go through one round of replication. But they have different rounds of division. So mitosis has one round of division while meiosis has 2 rounds of division. And I don't, this one of those is, I'm not reading through the answer choices, but it's this one. So mitosis has 1 round of replication, 1 round of division, while meiosis has 1 round of replication, 2 rounds of division. So basically everything I just said, I just didn't read to figure out which one to circle for y'all. At the end of telophase 1, the resulting daughter cells are, this one always tricks people up. Yeah, I'm sorry about the timer, Miss Pratt. I think I just kept the same time on all of them, no matter how long the answer choices were. Just practice anyways, right? So at the end of telophase 1, the cells are actually haploid. So what happens during meiosis 1 is that the homologous chromosomes are separated from one another. And so the homologous chromosomes are what make it diploid. So you have one maternal and one paternal. That's, of course, your diploid, your 2 sets of chromosomes. And so since we've separated the maternal from the paternal of each of our chromosomes, you only have 1 set in each of your daughter cells. They are duplicated, so they do have their sister chromatid, but the chromosomes themselves, the cell is actually haploid. Ooh, y'all got that right. I'm so proud of y'all. You're rock stars. So which following resulted from non-disjunction? So that means you have to figure out what non-disjunction is, and then figure out which of these is talking about. I wonder why my question box goes away so fast. I think it's maybe the update, because at one point that question stayed up the whole time last time when we did the game like a month ago. Okay, so non-disjunction has to do with when your sister chromatids or your homologous chromosomes do not separate during anaphase, and it's only going to occur to one of your chromosomes. So any of them that talk about like 3 pairs of a single chromosome, so 3 pairs of chromosome 21, and that is actually a disorder or syndrome called Down syndrome. It's actually called trisomy 21. So on March 21st, you used to celebrate Down syndrome awareness day, and we rock your socks out that day because it's 321. Haha. Okay, so our next question. So when observing a karyotype, how can one determine that a case of non-disjunction occurred? So if you look at the karyotype under all of the different chromosomes kind of lined up from longest to shortest, how would you be able to look at it and see that non-disjunction occurred? Okay. So having 2 sets of all your chromosomes is a normal individual, and so that's not showing non-disjunction. Having 2 sets of the X chromosome is if you're looking at genetically what a person's sex is. So that would be a female, and then one X and one Y would genetically show that they are a male. So the only one we have here that would be logical is this one saying that there's only one autosomal chromosome. So in the last question, we saw that there were 3 sets of or 3 of a certain chromosome. Versus here would be the other option, which would be that it would be missing a chromosome. So what we see with non-disjunction is either the cell will have 1 extra of a certain chromosome or will be missing one of its chromosomes. And so that's the result you see there. Nice job, y'all. You are rocking this. Why do I keep explaining it? Y'all got all these answers. What happens to the additional X chromosome found in female cells? So we know that males are perfectly fine having only 1 chromosome of X, like 1 X chromosome, because they are XY and they're perfectly normal, perfectly fine, no issues. So females actually have 2 sets of those X chromosomes, or they have 2 X chromosomes. So what do they do with that X chromosome? Well, they actually deactivate it and they store it as a bar body. So it kind of, they methylate it, they add a whole bunch of methyl functional groups to it, which is going to cause it to supercoil, which will deactivate it so it cannot be read. And then that little ball of genetic material will just kind of sit in the nuclear envelope. Nice job, y'all. And I think that is our last question there. Yes, I would like to exit the quiz. So how about we go over to heredity then? Because y'all didn't answer last time when I asked which one y'all want to do. So we're just going to select for you. We're going to go over to heredity, which is a little bit of math. So hopefully I made the math easy for you, but we'll figure out if I actually did or didn't. So our code now for our next game is 324324. What an easy code. Just the same code, same. So we have 324324 for our game if you're joining our next game. Remember, I can't press the start button unless somebody joins. How y'all doing today? Is this going okay? Y'all doing good? Y'all enjoying the game so far? Is this helpful? Boop, thank you. I see Miss Pratt's having fun. Anyone else having fun? Is this helpful for you? Yes, no, could be so. Okay, well, if anyone wants to join, they'll join us. 324324. Yay! Emily says she's having fun. Emily, are you joining this game too? I need to send out better reminders to my class to get them on here. You actually don't have to if you don't want to. I was talking to my husband about, like, if nobody showed up, that I was probably not going to do these anymore. Because I don't really know how, like, if y'all like doing them. Like, I got to figure out. I got to get the thing back to the right screen. Is it the right one? Yeah, I think that one's good. Okay. So in a monohybrid cross of complete dominance, what is the phenotypic ratio? Okay, so if we're looking at a monohybrid cross, and we're looking at complete dominance. Complete dominance means that it has, if you have one dominant allele, it will completely mask the recessive, and so you will only see it if you have one dominant. You'll see the dominant phenotype. And so since we're looking at a monohybrid cross, it means that they're both heterozygous for that one trait. So you should see a 3-to-1 ratio. Ooh, monohybrid, y'all. Monohybrid. We did not say dihybrid. Don't get tripped up on those. So, in a monohybrid cross of incomplete dominance, what is the phenotypic ratio? Okay, so this is really kind of monohybrid. Again, they're heterozygous for one allele. And so we'll see that one of them will be homozygous dominant. So it would, if we look at, I don't know, red versus white, we'll have one that's red, and then you'll have 2 that are heterozygous. So those ones will be pink. And then you'll have one that's homozygous recessive, which will be your white one. So it should be a 1 to 2 to 1 ratio. Oh, you're welcome, Miss Pratt. So we're looking at in a dihybrid cross of complete dominance, what is the phenotypic ratio? Okay, so in dihybrid cross complete dominance, what is the phenotypic ratio? So again, we're looking at dihybrid. We're looking at complete. So if it has at least one of those dominant alleles, it will show that dominant trait. So we should see a 9 to 3 to 3 to 1 ratio because we're looking at heterozygous for 2 traits. Nice job, y'all. So, in a population of 100 offspring, how many would you expect to be homozygous dominant in a cross between a homozygous dominant and a homozygous recessive? So, if we see that those parents, one of them is homozygous dominant and the other is homozygous recessive, you would expect 100% of those offspring to be heterozygous. So the question says, how many of those offspring would be homozygous dominant? The answer is 0. Because the fact that all of the offspring should be heterozygous. So, in a population of 200 offspring, how many would you expect to be homozygous recessive in a cross between a heterozygous and homozygous recessive? Okay, so if we're crossing between a heterozygous and a homozygous recessive, you'd expect the 50% of them to be heterozygous and 50% of them be homozygous recessive, because the fact that heterozygous has 50% chance of passing on the dominant or the recessive, and then the homozygous recessive has a 100% of passing on that recessive. So if 50% of them are going to be homozygous recessive, 50% of 200 is 100. So that's what your answer is. Nice job, y'all. So in a cross between a white recessive and red dominant, what is the probability of a pink offspring? I assume that I was probably doing incomplete dominance with this. And so, well, actually, if we're doing incomplete dominance, then it would be 50%. Well, yeah, no, 100%. But if we're doing complete dominance, it would be 0. Yeah, I was going incomplete dominance on this. So you should have had, no, wait, what was I doing? Cross between a white and red. That would be 100% would be red. So then you would have, no. What was I doing on this? Oh, no, that's what I was, no, what was I doing? This may have been one of those that I made in the middle of the night, and I have no clue what I was thinking. It was red. I don't know what I was thinking on this. I'll have to come back to y'all later on that one. Sorry, y'all. So we're doing red versus white, and I'm assuming this is going to be complete dominance. Then we're going to see that 3/4 of them will be red, and then 3/4 of them will be axial. So it should be 9/16. Nice job, y'all. I've got myself all worried that I did other crazy questions. In a cross between an individual with A blood and an individual with B blood, what's the probability of offspring of O blood? Okay, so A blood is going to be IA with a recessive little I. And then B blood would be IB with a recessive little I. And so since each of them can pass on that recessive allele, and there's a 1/2 chance of each of those, 1/2 times 1/2 gives you 1/4. So it should be 1/4. There we go. That's an example also of codominance for that one if they were AB blood. So cross between 2 roan cows, it's probably of a white cow. So this one shows incomplete dominance. Okay, so if we're looking at 2 roan cows, that means that we have 2 heterozygotes. And so the probability of being homozygous recessive is 1/4. Nice job. And that should be, I think, oh no, we have one more question. Which the following description about dominance is correct. So incomplete dominance is going to involve where neither one is completely dominant. So you see a blending of the two. And codominance has to do with that you're going to see each of those dominants independent of one another. So you would see like an AB blood type, you would see A and the B glycoproteins. So the answer should be incomplete dominance involves heterozygote goat resulting in an intermediate between the 2 dominant traits. Nice job. And that should be the last question in the game, leading us to our one and only last game left, which is cell communication. I'll make that smaller. There we go. So 645-706, and that's the last game that we have today. Ooh, and we have somebody already in. Yay! Y'all are the best. Can someone type it into the question box so that it will have it for if anyone comes a little late? So 645-706. 645-706. Please. Yay! Thank you, Emily. Okay, I'm going to press the go button. I think I'm going to have to adjust the screen again because I think I messed it up when I was trying to get the code up for y'all. There you go. Okay. So where does the ligand bind to initiate signal transduction? So the ligand is going to bind to the receptor, and where the receptor is kind of varies based on the type of ligand. If it's a polar ligand, it'll be on the membrane, and then if it's a non-polar ligand, then it will be inside the cell because it can pass through the membrane. So nice job, y'all. So where would you find a receptor for a steroid hormone? Y'all, my setup for this thing is so funny. My laptop's sitting on a laundry basket, and my phone is propped up using high-heeled shoes. Okay, so where'd you find the receptor for a steroid hormone? So we have to remember that steroids are non-polar. And so because of the fact that they're non-polar, we would find it in the cytoplasm. So in the cytoplasm because steroids are non-polar. Which of the following is a secondary messenger? So, secondary messenger would be cyclic AMP. Another secondary messenger is calcium, so Ca2+. ATP is not a secondary messenger. It is a energy vehicle, so it drops off energy different places. Protein kinase is responsible for phosphorylating, and then the ligand we already said was a signaling molecule. So which of the following is the order of steps for a signal transduction pathway? So in terms of signal transduction pathway, our first step is reception. That's when the signal is received by the receptor. We then go through transduction, which is how the signal message is going to be amplified and transmitted from the receptor to the response. And the final step is, of course, that response. Nice job, y'all. How are steroid and protein hormones different? And thankfully, all the answers are on your screen instead of you trying to read my screen. So we saw earlier that our steroid hormones are going to be nonpolar, so they're hydrophobic, while our protein hormones are going to be polar, so they're hydrophilic. Did I say? Okay, y'all. Steroid hydrophobic, protein hydrophilic. I think I may have said the wrong word a second ago. I know my content, I'm sorry. So which of the following is a potential response for the cell? Okay, so if we're looking for a potential response for the cell, a conformational change to the receptor is called reception. A transcription factor is going to bind to DNA to turn on or turn off a gene, so that would be some type of response that could happen. Phosphorylation cascade is part of transduction, is where we're going to send the message by basically phosphorylating using protein kinase. And then releasing calcium from the smoothie R, that's a secondary messenger, which is also part of transduction. So the answer is transcription factor. Ooh, we got somebody get it right. Nice job. How is paracrine and hormone signaling different? So our hormone signaling is going to take place in our bloodstream. So it's going to be released by exocytosis into extracellular fluid, the interstitial fluid, that will then move into the bloodstream and travel all around the body. Versus paracrine signaling is going to be kind of local signaling to nearby cells. And so it should be right here. Hormone signaling involves bloodstream, long distance, and paracrine involves extracellular fluid and short distance. I know that was a little long for you to try to read. Sorry. How are ligands cell specific? So each of our cells is going to have different receptors that will bind that ligand. So as long as the ligand can bind to the receptor, then the cell will take in that ligand. So nice job, y'all. So describe the phosphorylation cascade. So we mentioned this earlier. The enzyme that's responsible for phosphorylation would be our protein kinase. Protein kinase is going to phosphorylate a protein, a relay protein, within the phosphorylation cascade, within that, pathway. And so that phosphorylation will cause a shape change, which activates it or primes it to do work. Phosphatases are going to dephosphorylate. So this is how you would stop the message. So it would take off that phosphate molecule, which would kind of reset the dominoes in a way. ATP does add phosphates, but not to the relay molecule, and the mitochondria is responsible for ATP synthesis using oxyphosphorylation. So more than one ligand can bind to the same receptor. Okay, so in this case, we see that it is true because of the fact that if they have the same shape, then they can bind to the same receptor. This is why you can see like morphine can bind to the same receptor as endorphins within the body because they have a similar chemical structure, so they bind to a similar receptor. That was the last question. Okay, so which means that is the end of my stuff that I have for you. So just as a quick reminder, if you go to apbiopenguins.weebly.com, you have access to these games as well as other games. So I've got them broken up by each of the units, as well as I've got like overall just CED games and then just unit review games and just extra games. So all these games are available for you as well as everything else that I have for the review. So I've got all the TikTok games, like all of my TikTok content's on there. I've got FRQ Fridays from last year. I'm planning on maybe trying to see if I can build some more of those. I've got all my marker learning sessions, all the daily review that I've been posting. I've got my Instagram live sessions that I've done with my students this school year is on there. So like everything that you would possibly need that I could give you to review is all there, including my 315 page review guide that breaks everything down for you. So if you don't have any questions and there's nothing that you need, I'm going to sign off and go spend the rest of the day with my kids. So y'all good? Anyone need anything before I log off? Going once? You're welcome, Emily. I hope this was helpful. Going once. Going twice. Goodbye, y'all.